Hello everyone, welcome to a new session on metabolism. In this presentation, we will be looking into in detail the hexose monophosphate pathway or hexose monophosphate shunt. Okay, now why it has been given the name hexose monophosphate? Because the first substrate to initiate the HMP pathway, it is otherwise known as HMP. Okay, HMP pathway, it is the glucose 6 phosphate, right? Glucose 6 phosphate, glucose is hexose, and 6 phosphate, there is only a single phosphate, there is only a monophosphate on the sixth carbon of glucose, isn't it? So it is a what you call hexose monophosphate. In a cell, in a biological system, we can see there's a hexose monophosphate pool, which is composed of glucose 6 phosphate, fructose 6 phosphate, glucose 1 phosphate, etc. So here in this particular HMP pathway, we can see glucose 6 phosphate is the first substrate to undergo the reaction or the pathway, and we can also see the fructose 6 phosphate being produced as one of the final products in the pathway. Okay, it is otherwise referred as pentose phosphate pathway. That is, in between the phosphorylated pentoses are produced and play a very important role in running the HMP shunt. Okay, that is, you can see here the ribose phosphorylated ribose is being produced, ribuloses and celluloses. So all these are very important pentose phosphates in the HMP pathway. Okay, phosphogluconate pathway, yet another name for the pathway, because the first phase of the HMP pathway, it can produce, or it produces phosphogluconate as a intermediary substrate. Okay, then major contributions to the elucidation of various reactions in the HMP pathway was done by Warburg, Lippmann, Dickens, Horaker, and hence it is also named after these scientists. Okay, it's the Warburg, Lippmann, Dickens, Horaker shunt. Okay, now the pentose phosphate pathway, it is otherwise it is considered to be an alternative pathway to direct glucose oxidation. That is, it is an alternative pathway to glycolysis and TCA cycle for glucose oxidation. And it occurs in the cytoplasm, in liver, adipose tissue, the adrenal cortex. You can see it in the lactating mammary glands, thyroid glands, erythrocytes, eye lens, and such kind of organs. But it is this hexose monophosphate pathway is not found to be operating in non-lactating mammary glands and skeletal muscles, right? So now the next unique feature is that no ATP is produced. Even though it is considered to be an alternative pathway to glycolysis, HMP pathway doesn't produce any ATP. But the unique thing is, like, it produces NADPH instead. Okay, NADPH, and NADPH is mainly produced through hexose monophosphate pathway. Okay, NADH is produced, ATP are produced, every other FADH2, etc., produced in various glucose oxidation pathways. But NADPH, it is produced only in hexose monophosphate pathway. These HMP pathway, it operates in, as already mentioned, in cytoplasm, and it supplements the glycolytic pathway, and nearly almost like 10% of the glucose entering the biological system enter the HMP pathway. And we can see that it is a major source of NADPH, and it occurs in those regions which require NADPH for the biosynthetic processes. For example, the NADPH, it is required for the biosynthesis of fatty acids and cholesterol, the steroid, and hence you can see NADPH is produced via HMP pathway in adipose tissue and liver, right? It is also involved in the detoxification of chemicals, like detoxification of drugs and poisons in liver, neutralizing ammonia, etc. Where does it take place? It takes place in liver and adrenal, what you call kidney, especially in liver. Okay, so these are the HMP pathway helps in providing the sufficient amount of NADPH for all these reactions. It also helps in the synthesis of fatty acids, cholesterol, and also the amino acids that involve glutamate dehydrogenase. Okay, we can see that erythrose phosphate, which is an intermediary substrate formed in the hexose monophosphate pathway, is required or is necessary for the synthesis of amino acids like tyrosine, tryptophan, phenylalanine, right? So all those aromatic the amino acids are being produced with the help of NADPH, right? And the pentose phosphate pathway. So again, it is a very important pathway for various biosynthetic. Okay. Then you can see that it also produces, that is the what you call the pentose phosphate pathway. It also helps in the synthesis of pentoses, especially phosphorylated pentoses. Now, what is the function of pentose phosphates? Pentose phosphates are used for the synthesis of nucleotides as well as the nucleic acids. Okay, for example, ribose 6-phosphate is an intermediary substrate in HMP pathway. Okay, this ribose 6-phosphate it can be used for the synthesis of various kinds of what you call coenzymes which involve the nucleotides like ATP, what you call NAD, FAD, etc. And also it can be polymerized used for the polymerization and synthesis of nucleic acids. Okay, so it do have a very important role, the HMP pathways. Okay, now we'll see the phases. As already mentioned, the HMP pathway, it can be divided into two phases, that is the oxidative phase and the non-oxidative phase. Okay, the oxidative phase, it starts with glucose 6-phosphate, and this undergo the oxidative phase to produce ribose 5-phosphate or ribulose 5-phosphate or cellulose 5-phosphate. Okay, and then these products, it will enter the ribose 5-phosphate and cellulose 5-phosphate. It will enter the non-oxidative phase. Okay, and that will result in the formation of different kinds of pentoses and hexoses, phosphorylated hexoses. Okay, so we can see the oxidative phase is mainly concerned with the production of NADPH and carbon dioxide. Okay, it also produces the pentose phosphates. These are pentose phosphates. Fine, and here we can see the hexose, the glucose 6-phosphate, it is getting converted into pentose phosphate. Okay, that is glucose 6-phosphate, which is a hexose phosphate. It is getting converted to pentose phosphate in this oxidative phase of the HMP pathway. In the non-oxidative phase, this is like represents the non-oxidative phase, and here what happens is this involves a series of reactions by which pentose phosphate, okay, this is a pentose phosphate, the pentose phosphates, they are converted to intermediate products of glycolytic pathway like glucose 6-phosphate or fructose 6-phosphate or glyceraldehyde 3-phosphate. So all these are intermediate substrates of glycolysis, isn't it? So here now in the non-oxidative phase, you can see the pentose phosphates are converted to intermediate substrates of the glycolytic pathway. Okay, and these intermediate products can enter the what you call glycolytic pathway, or it can be channeled back to the pentose phosphate pathway. Okay, either way it can enter the what you call cycles. Okay, now we can just look into in detail the oxidative and the non-oxidative phases. Okay, in the oxidative phase, the first phase is dehydrogenation of the glucose 6-phosphate. The glucose 6-phosphate, it undergo you can see here, yeah, oxidation. Okay, we can say it undergoes oxidation to produce 6-phosphogluconolactone. This particular reaction is catalyzed by NADP dependent glucose 6-phosphate dehydrogenase. Okay, this NADP, it is an oxidizing agent. It undergoes reduction to produce NADPH. That's the first NADPH molecule to be produced here. Okay, the 6-phosphogluconolactone, it undergoes hydrolysis. Okay, water is added and it undergoes hydrolysis to produce 6-phosphogluconate, right? And this reaction is catalyzed by the enzyme lactonase. Even this particular reaction can even take place without the help of the enzyme, but the reaction rate will be slow. Okay, so lactonase, it hydrolyzes 6-phosphogluconolactone to produce 6-phosphogluconate. Okay, then this one, the 6-phosphogluconate, it undergo decarboxylation, oxidative decarboxylation to produce ribulose 5-phosphate. Okay, so this will the 6-phosphogluconate, it undergoes you can see dehydrogenation. The hydrogen which is released from here, it will be taken up by the NADP and get converted to NADPH. That's the second molecule of NADPH being produced. Okay, and decarboxylation, it will result in the release of a carbon dioxide molecule. Okay, since a carbon is released, what happens? Till here it is a 6-carbon compound, isn't it? Here you have 6 carbons in the chain, right? But when one carbon is released as carbon dioxide, what happens? It will become a pentose. Okay, so the 6 carbon 6 gluconate, it is getting converted to 5 carbon ribulose 5 phosphate by decarboxylation and oxidation. This catalyzed, the reaction is catalyzed by 6 phosphogluconate dehydrogenase. Okay, as already mentioned, it is an NADP dependent enzyme, and it also requires magnesium or manganese ions, the presence of these ions. Okay, now here you can see the aldohexose phosphate. Okay, this is the aldose, phosphoaldose. Okay, it is a aldohexose, phosphorylated aldohexose. It is getting converted to phosphorylated ketopentose. Ribulose is a ketose. You can see here this is a keto. Okay, a phosphorylated aldohexose gets converted to phosphorylated ketopentose in the oxidative phase. Okay, and later this you can see that the ribulose 5 phosphate is the one which actually enter the what do you call the next phase. But before that, it can take up interconversion. Okay, the interconversion of the pentose phosphates, that is ribulose 5 phosphate, it can undergo reversible, you can see it is a reverse reaction, reversible isomerization to produce ribose 5 phosphate. Okay, it results in the formation of ribose 5 phosphate, which is a aldopentose. You can see it is aldose, right? CHO, right? This ribulose 5 phosphate, it can also undergo epimerization to produce cellulose 5 phosphate. You can see the cellulose 5 phosphate. Okay, now so ribulose 5 phosphate, it can undergo epimerization resulting in the formation of cellulose 5 phosphate, and the reaction being catalyzed by pentose phosphate epimerase, while it can undergo isomerization to ribose 5 phosphate, the reaction being catalyzed by phosphopentose isomerase. Okay, now in a reaction, you can see here in a what you call HMP reaction, 2 molecules of cellulose 5 phosphate and 1 molecule of ribose 5 phosphate are required for to take up the non-oxidative phase of the HMP pathway. That is, to pass on to the next step, they need the formation of 1 molecule of ribose 5 phosphate and 2 molecules of cellulose 5 phosphate. Okay, so this is where the oxidative reaction stops. Now it starts the non-oxidative reaction. That is, here NADH is not being produced. That is it. Okay, in this particular phase. Now